really quickly, last class we did a Socrative quiz. A gene that's being expressed will most likely be in euchromatin or heterochromatin, was the question. So euchromatin is the type of chromatin where the DNA is loosely compacted and transcription occurs. So a gene that's expressed will be in euchromatin, not usually, this is not a 100% rule, not usually in heterochromatin. So all of these are about genes being expressed, genes being actively transcribed. So, transcribed gene. CpG dinucleotides will be methylated or not methylated. Addition of methyl marks in the promoter on the cytosines prevents transcription factors from binding and starting transcription. So if the gene's being transcribed, it's going to have methyl marks or no methyl marks. So they're not going to be methylated. So gene expression, no methylation. Likewise, the gene that's being expressed, we're talking about histones now in the third question, the proteins that DNA is wrapped around. The histone tails get acetylated or methylated. Genes are being transcribed. It's acetylated. So again, the common pattern, if there is any rule, which there isn't, but if there was a rule, it would be that acetylation of histones is involved with active genes, and methylation of DNA is involved in turning off gene expression. So methylation of DNA, genes turned off. Acetylation of histone tails, transcriptions turned on. Any questions about this? So general patterns. If I asked you for a trend, that would be the answer. There are some rare exceptions. Then at the end of last class, I asked everybody to participate in a mock exam to try to get you recalling information from earlier in the semester to start preparing for the final. So I want to go briefly through the questions of what I'm going to do is I'm going to show you which was the correct answer and which was the most commonly chosen incorrect answer. So I've selected these few questions that I'm going to show you, not all of the questions from the exam, just a few of them. These are the questions that most of you got wrong. So if I look through every question, and most of the questions, most of you got right. But there are a few questions where there was still clearly some confusion. Or they were hard questions, and there is no confusion, it's just hard. So, question 3. The most commonly chosen incorrect answer, MCI, was A. That if there's a disease that affects women and not men, it's likely caused by a gene on the X chromosome. So that's not the correct answer. That was the one that a lot of people chose. So with that ruled out, which one is the correct answer? It winds up being C. Okay. This is not something we discussed in class, which makes it a good segue to talk briefly about why C is the real answer. So can anybody think of a scenario where C is true? How could you have, or why 3B is a possible answer, in other words? So most of you said it was answer A. So in what scenario could B be true? That a mutation in an autosome could cause a disease that only occurs in females? It doesn't require genetics information. There's a pretty simple answer. There's a really simple answer. Okay, you're thinking too high. Think even simpler than that. No, that's too low. Come back up a little bit. Okay, let me lead a little bit if I may. Do I have a cervix? I'm not talking about a neck. I'm talking about the genital version of the cervix. Do I have a cervix? This is not a trick question. No, I do not have a cervix. You didn't know that either. It hasn't been covered in class. You're not required to know that for the final. Right? So am I going to get cervical cancer? No. Cervical cancer is a disease that can only occur in women. If there's a mutation in a gene, it could be on an autosome. If there's a gene that has a mutation that predisposes you to getting cervical cancer, that is something that will only happen in women, because women are the only sex that have cervixes. So there could be a gene on an autosome, not an X chromosome in other words, that controls whether or not you get cervical cancer. But that depends on you actually being the correct sex to have that tissue to get the cancer in. I thought about leading with breast cancer because it's maybe more familiar to everybody, but men get breast cancer too. So that's a bad example. Right? Both sexes can get breast cancer. I figured only one sex could actually get cervical cancer. So that's why without additional information, we can't tell. So it might be that there's a trait that's limited to one sex, not because of where the gene is located, sex chromosome or not, it's because there's a difference between the sexes. Can women get Adam's apple cancer? Presumably not, because most women don't have one of these bony things in their throat. So that's the concept there. Any questions or follow-up issues with that? So it's a little bit misleading question. We hadn't covered that in class. So cystic fibrosis. How many, so this is pedigree analysis. Give some information about the family. We want to know what the probability of a child being a carrier is. So most of you thought you got this right. In fact, this is the answer that I came up with when I first tried this. B was the most commonly chosen incorrect answer. It's not a half. And the vast majority of us all chose this answer when we did this, including me. So I had to think about it. There was no teacher guide to this question, so I had to really think about it. And then I think I figured out why the answer is something else. So here's, say, the family. How do I fill in the phenotypes here? Normal couple has a child with cystic fibrosis. And we know that this is recessive. So what are the phenotypes? How would we fill in the symbols of the parents? Are they clear? Are they half filled in? Or are they totally filled in? Why half? So you've got one kid that's got the disease and one that doesn't. That's what this question says. The first child has cystic fibrosis, the second child's unaffected. It doesn't matter what the sexes of the kids are. So if there's a recessive trait and one kid has it, what does that tell us about the parents if they're both unaffected? They're both carriers. Boy, my spelling is horrible. So if this kid is unaffected, could either be, what are the genotypes possible here? So each of the parents is plus over minus. They have one kid that's minus over minus, has the recessive disease. What are the possible genotypes of this kid? So it could be plus over minus or plus over plus. Because each of the parents has a plus to contribute, each of the parents has a minus. It's almost like what you did was you did the Punnett square. Each parent has a plus and a minus. So if you fill in the Punnett square, you have 4 different options of genotypes you could produce in the kids. The only time when you get the phenotype is when you're minus over minus. So that's that case. So what's the probability that this second kid is a carrier? Right? 2 out of the 4 boxes over here are the carrier heterozygous genotype. That's why everybody answered 50%, half. You've got a 1 in 2 chance of being carrier, except what else? So those are the carrier genotypes. What do we know about this individual's genotype? They're not affected by the phenotype. So what does that tell us about their genotype? Which of these 4 genotypes in the Punnett square are they not? Can't be minus over minus. So that rules out one of the 3 squares in the Punnett square as a possible genotype for that individual. So what's the probability that they are heterozygous? 2/3. And not a single person in here, or maybe one of you, I think one of you said 2/3. Which is, again, fine. I didn't do this right the first time I did it either. But now you know. So you've had, it's a trickily worded question. So yes, 50% is trickily. It's a trickily worded word. So the individual could be, well, let's see. So why was the individual not plus over plus? No, they can, but the question is asking specifically about if that individual is heterozygous. So there are 2, they can't be minus over minus. So they're either plus over minus or they're plus over plus. So that's 2 out of 3 of the possible genotypes are carrier. If they had asked what's the probability that they're plus over plus, and that would be 1 over 3, 1/3. Yeah, good to clarify. Questions 11 and 12. So most commonly incorrect were B for question 11 and D for question 12. All right. So if we make these mutations, the first, question 11 is tricky. And I know I didn't give you much time to do this, and some of you might have just done A, and turned it in, which is fine. I didn't see anybody actually did that. You might have, and I didn't miss it, or I missed it. But so this one would take a little bit more time if you wanted to answer this one really carefully. So if we make a G to A mutation at position 53, that's right there. So if we turn that G into an A, why does that turn this coding region into a shorter something? So a lot of you answered that this was the correct answer, B, G to A at position 53, like I've drawn up here in blue. So what's the basis for that response? What did you do by making an A instead of a G?
It creates a stop codon, but it's looking at mRNA versus.
Right, so everybody shows evidence of understanding what's going on. This creates a stop codon, a TGA, and it's even in frame. Because this question said you start with the first AAT, that's an amino acid, so you see GAA, TGA, stop codon. But what's the question asking? It's asking about whether or not the messenger RNA is going to be shorter. Translation has nothing to do with transcription, which is what produces the messenger RNA. So if you're thinking translation, that's a step beyond what happens in the messenger RNA. Stop codons, that's past this question. So that's why it's none of the above. None of these mutations will affect transcription. These are mutations that are changing amino acid sequences. That's translation. That's down, right? DNA replication, then transcription, then translation. What would you have to do to make a shorter than normal messenger RNA? What sort of mutation would cause a short messenger RNA, shorter than normal? What regulates the length of a transcript? How does RNA polymerase know where to stop transcribing? There's a particular DNA sequence that RNA polymerase reads across that says, hey, this is the end of the transcript, essentially. So that's the poly A signal sequence. Right? At the end of every gene, downstream of where the codons are, way past that in the 3' untranslated region down here, there's an AAU, AAA. RNA polymerase transcribes that, and then about 20 nucleotides or so downstream of that signal sequence, it stops transcription. the poly A tail gets added. So the only type of mutation that I can think of offhand that would create a shorter than normal messenger RNA is what would have to happen up here somewhere. Let's say over here to the left in the 5' end of a transcript, there's a sequence that's A. Let's say you have 6 A's in a row up towards the start of the transcript. What sort of mutation would cause a shorter than normal transcript? What could you do to those 6 nucleotides to make transcription stop earlier? Yeah, so if you had a mutation that changed that third A into a T in the DNA sequence, that would create a premature and early poly A signal. RNA polymerase would hit that, you say, oh, that must be the end of the gene. So it's really critical, and ask questions now, come to office hours. It's really critical to be able to separate the processes of transcription and translation. Question 12 then. Now we're talking about what you were expecting. What mutation is going to cause a premature termination codon, a premature stop codon? So a lot of you said none of the above. Not A, not B, not C. So in the second version of the messenger RNA sequence, so let's see. Look over this again. Let's first, as I always do when I'm looking at mutations, underline all of the codons. So if we change position 59, this U, to a C, does CCC change anything? Is that a stop codon? That's option A, T to C at position 59 right there. That's CCC, proline, I think. It doesn't matter. It's not a stop codon. What if we turn that A into a G? That's option 2. GA. It's an amino acid. It's not a stop codon. So what about insertion of a G after the G at position 54? So if we stick a G in there, then in a different color we've got GGA as a codon, GCC, and then UGA, the stop codon. So the right answer there was C. One more question. 8. Oh dear, what did I do? It's been a long time since we thought about meiosis and drawing chromosomes. Most commonly incorrect answer? Almost everybody, it's kind of split. Most of you thought it was Sue's fault. I don't know if that's some sort of a tendency to blame it on the woman, which is totally untrue. But so the most commonly chosen incorrect answer was Sue. Although there were almost as many people that said Bob, and almost nobody said C. You can't tell from these genotypes whose gamete it was that's at fault for creating a stillborn son that has this genotype. Triploid, trisomy for one chromosome. Why isn't it that we can't tell? Right, so both of the parents have at least one capital A allele. So it's possible, and I'm prepared to draw this out if you want, or we could maybe talk about it in office hours. I'll record it, and then I'll post it, and everybody can watch it if they want to. But we can go through and show that there could be errors in meiosis in either parent that would lead to this sort of a genotype. So it's relatively unimportant that Sue happens to be homozygous for big A. That doesn't make her any more likely than Bob, poor Bob, to have caused this problem. So briefly, what is the problem? How do you create a child that's trisomic, that has 3 copies of a chromosome? What did fertilization look like? Just quickly, half a minute, we'll be done. We'll get into new content. What are the genotypes that are necessary to produce a trisomic individual? How would I put in these 3 A's? How do you divide those among the 2 gametes? So we could put two A's in the oocyte and one big A in the sperm. That's one possibility. That would be Sue's fault, because she was the one that produced the oocyte. Or the opposite. You could have an oocyte that's haploid like it should be, and a sperm that has an extra copy of the big A. That would be Bob's fault. Questions? Finally, we get to the last case study in epigenetics then. If there's no other questions, procedural questions, class questions, finals coming up, anything at all. Then it's on to asthma. So I closed last class by talking about how horrible our air quality is, which everybody's aware of. And I said that I had shown some data from last year or 2 years ago's State of the Air report. I found this year's, it actually was just released recently. And so, as you already know, so I won't belabor this point, our air sucks. And even though we're doing better now in Fresno, well, I don't know, I guess we're not doing any better. We've been 4th highest county or region in the US for air quality 2 years in a row. Woo! And so LA and Long Beach have been the worst. And then some of these are flipping, right? So Bakersfield went from 2nd to 3rd, but unfortunately that meant that Visalia, Porterville, and Hanford went the other direction. So it's just a bad place to live for quality. We're going down, this is where we went down. Sorry, I misremembered the data. We've gone down from 2015 to 2016. We were in 1st place last year. We're in 2nd place this year for short-term particle pollution. Wow. Put that on the Chamber of Commerce flyers that they mail out. Come to Fresno, we're now only the 2nd highest air quality. And then in year-round, so not just short-term, but also year-round, it's all bad. So there are a number of people, we have a lot of views, we have a lot of, 5th largest city in the state, huge population center, really bad air, which means a lot of people are affected by the air quality. And the air quality is caused by a lot of different factors, according to this report. As you might expect, it's all the idling we do at the stoplights because those stoplights on Shaw just suck. It's nice to have a regular stoplight every half mile in case you need to go the other directions, but otherwise, geez. So that brings us to research. And the whole point of today, the new material, is to be able to analyze and predict what sorts of epigenetic changes might be involved in the production of asthma. So we're going to learn a little bit of cell biology, we're going to talk a little bit about epigenetics, and mainly predict and interpret scientific data. So this is a study that, as I shared with you last time, is from Dr. Nadeau was the first author on this. It was published a couple of years ago. All of what comes next is from this paper. So Dr. Nadeau is a pediatrician who works at Stanford. And so we've got the, whatever the hell Stanford's, the tree, versus the Bulldogs, so we've got the better mascot anyway. This is a story that she told about pediatric asthma patients in Stanford versus in Fresno. So hopefully we win. And so we're not too far apart physically, but there's a big difference in air quality between Silicon Valley and the Central Valley. This is the cell biology you need to know to understand this, and it's not really that big a deal. This looks tricky, but most of this you can just gloss right over. You can see eyes glazing over already. A bunch of pathways and gene names. Here's the thing. There are a population of cells called Th2 or helper T cells that are helpers. The H is for helper. Th2 cells. When you have a lot of Th2 cells, that causes the symptoms of allergen attacks or asthma. So it's when your Th2 cells are active that brings on wheezing, constricted airways, and those sorts of things. This is part of our immune response. And so it's a useful thing to have allergies or allergic reactions. This is the body trying to fight off an invader. But we have to keep those systems in check too. Our bodies have to be able to regulate not overproducing Th2 cells. Too many Th2 cells, bad, in other words. So there has to be some regulation of those cells. Here are some genetic pathways. We got pointy arrows and blunt arrows. So which cells do we want to have to prevent hyperallergic reactions or severe asthma? Th17, Th1, Th0, Tregs. Right, Tregs. Tregs inhibit Th2 cells. So we want, if you were an asthmatic, what you want to do is have an increase in your T regulatory cells. They're regulatory cells because they regulate the Th2 cells. And there's an important gene that's not written on this plot, but I'll draw it on here along that arrow from Th0 to Treg, the FOXP3 gene. So along with these other genes, TGF-beta, IL-12, the FOXP3 gene is important in turning a Th0 cell, which is a cell who hasn't decided what sort of a cell it's going to be yet. Think of it sort of as a stem cell. It's uncommitted to its fate yet. Depending on which genes get turned on, those are the genes shown by all the arrows, that cell will be shunted off into one of a few different developmental pathways. It could turn into a Th17 cell or a Th1 cell. If FOXP3 gene, it's a transcription factor, right, a gene that's important in turning other genes on, like genes that turn cells into Treg cells, if FOXP3 is active, Th0 cells turn into Tregs, and that would help, presumably, people that are having severe allergic reactions or asthma attacks. And so Dr. Nadeau's hypothesis, as it says down here at the bottom, was maybe it's this gene, FOXP3, that's involved in severe asthma in the Central Valley. So that was her hypothesis. So all she did was she took patients from her clinics, pediatric patients. So there's a little key I've written here down below. There are 4 different classes of patient. You've got 2 different types of kids from Fresno, with asthma or without asthma. And you've got the same control cases from Stanford, kids with asthma, kids without asthma. Okay, so we're comparing 2 different geographical places, Fresno versus Stanford. And then within each of those geographical places, we're separating the kids into who's got asthma and who doesn't. So what's the prediction? Here we're looking at, she took samples of tissues from these patients. She wants to know how many of the T regulatory cells are functional in each of these groups of individuals. So basically, how well equipped is your body to stop those T helper cells from creating symptoms of asthma? Who's got the least functional T helper cells? Or Tregs, rather. Fresno, but Fresno plus. Fresno kids with asthma had the lowest functional, the least functional T regulatory cells. Who had the best T regulatory cells? Those punks from Stanford. Yeah. So kids from Stanford that don't have allergies, super good T regulatory cell function. So that's comparing Fresno with asthma, Stanford not asthma. What other comparisons should we be making? To figure out if there's a genetic component to this or an epigenetic component. What other comparisons are useful to make? That's just one. What do you see when you compare Fresno kids with asthma and without? What's the trend? Do Fresno kids without asthma have better or worse or the same T regulatory cells? So percent function in this group Fresno kids without asthma is higher on average than the kids with asthma. Statistically significantly different, or is just chance? So what does it mean? What is this p minus or less than 0.05 with this bar here? What does that mean? It's significant. What's this bar indicate?
And between those two individuals.
It's between these two groups. So what they've done is a statistical test comparing just Fresno with asthma, Fresno without asthma. Are those two sets of measurements significantly different? The answer is p is less than 0.05. Yes, there's a statistically significant difference. So what happens when you compare Fresno kids with asthma to Stanford kids with asthma? FA versus SA. That's this graph up here, or that's that comparison. Also p less than 0.05. So if you're in Stanford, in other words, your T regulatory cells work better regardless of whether you've got asthma. Benefit of living in Silicon Valley, maybe. But in both of these places, both in Stanford where the air is much better and in Fresno, if you don't have asthma, you have higher function in your T regulatory cells. Right? So this difference, right, Fresno asthma versus no asthma, and Stanford asthma versus no asthma, there's always an increase wherever you live. If you don't have asthma, this predicts, this says, I could tell you, if you live anywhere, if you've got asthma, I could pull some of your cells and I would find out that your T regulatory cells don't work as well. That's what these data suggest. Within one geographical region, if you've got asthma, your Treg cells don't work as well. So it's time to make a prediction. I've already told you these first bullet points. So FOXP3 is important in making T regulatory cells. And it's also really important, now I'm going to tie this into epigenetics, this third bullet point, that mouse studies showed that environmental factors, maybe like dirty air, can epigenetically modify the FOXP3 gene. And as we would have predicted from what we talked about, again, reviewed at the start of class, if that gene is methylated, the DNA is methylated, you would predict that it would not be expressed. DNA methylation causes gene repression usually, no transcription. So would you predict, don't answer now, think, will FOXP3, the gene, be more heavily methylated in A, Palo Alto versus Fresno kids, and then B, children with or without asthma? If you go to the next slide, I'd like you to draw a plot. Make a graph like the ones we just saw. There are 4 conditions down across the x-axis. Fresno with asthma, Fresno without, Stanford with asthma, Stanford without. What's the y-axis going to be? So what are you going to put on the y-axis? Draw your predictions and submit it and attach it to this Google Classroom assignment I just sent out. Take about 4 minutes or so. I'll circulate and answer questions if you have any, and then we'll come back together and